Hi everyone, my name is Sahil Agrawal, and on behalf of the entire team of PENdulum Global, I wanted to welcome you to our webinar training video on the biology of HIV infection. So you may be wondering, why are we spending some time on developing a biological and a clinical understanding of HIV infection when in reality we're going to be focusing on tackling the social stigma associated with HIV through our PENpal program? Our reasoning is twofold. Firstly, understanding how HIV infection occurs will help us find out why the social stigma exists for HIV in the first place, something that we're going to be going into a lot more detail into later on in our video on the psychosocial aspects of HIV infection. Secondly, we're hoping that we can give you a comprehensive understanding of HIV so that you can become more aware of all of its aspects and perhaps share that awareness with your peers on your campus. If you're an official volunteer, you have already received the training packet. It will be found below this video if you're on our website, pendulumglobal.org. And this is basically a fill-in study guide and a set of questions for you to answer as you watch the video. As I wanted to clarify, you're not going to be graded on your performance on this packet, but it's really just a way for you to be an active listener and to retain the main points of each of these videos. So without further delay, let's begin our exploration into the biology of HIV infection. So I'm going to try to make this as interesting as possible. I'm a very visual learner, so I'm going to be spending a lot more time showing rather than telling over the course of this biological video. So let's first consider a healthy human body. The body's primary defense against infection and conditions like cancer is the immune system. The immune system is made up of a series of cells that work together in a complex mechanism to keep the body healthy and without disease. Let's go over some of the cellular players of the immune system. So without going into too much detail, the key cells in the immune system are known as white blood cells, which the scientific world calls leukocytes. They're created in a number of different body sites, most importantly the bone marrow, and are found primarily in the lymph nodes of the body, as highlighted in the image on the left. Generally speaking, there are two major types of leukocytes, and you already might be familiar with them. The first are the phagocytes, which engulf foreign organisms, and the second are the lymphocytes, which help our bodies remember foreign invaders so that we don't get sick again. So now that we have a little bit about the basics of some of the key cellular players of the immune system, let's talk a little bit about what the immune response looks like. And this is a figure that I found from West One Services. And I kind of rotated it on its side so that the numbers are a little bit skewed, but it's a good image that kind of gives an overview of what the immune response looks like in response to some sort of foreign invader. I'm going to go through the first few steps because really that's what's going to be critical for us to understand where HIV creates a little bit of an issue or some sort of a stop in the immune response. And the rest of the steps start to get a little bit gray and confusing later on, so I'm going to kind of stay away from those because I want you to get a good general overview. So when a foreign substance or a molecule called an antigen enters the body, a series of cells will recognize that antigen as foreign and respond by triggering a set of lymphocytes known as B cells to produce antibodies that attach to the antigens and mark them for destruction. And this destruction can occur through an entire cascade of proteins and other cells, including a key player, the T cells. This protects us from disease or provides us with what's known as immunity. Just a little bit of a fun fact for those of you who are interested in biology and medicine. The reason why we call them antigens is because they were formerly known as antibody generators, antigen. And so you can see that I've listed some of the crucial steps of the immune response here, particularly pointing to steps 1 through 3 in the cascade. And so what I've done in this slide here is I've blown up a portion of the previous figure in order to just focus on the T cells. And the reason why I've done that is because the T cells are crucial to learning about HIV infection. So I wanted to spend a little bit of time giving you an overview of the different kinds of T cells that exist within the immune system. Once again, T cells respond to the antibodies that have marked certain antigens for destruction. There are 4 different kinds of T cells that are produced by the body in response to those antibodies to fight off the infection. Killer T cells, as their name describes, destroy the antigens. Helper T cells are going to promote the phagocytosis for the engulfing of foreign invaders. And they're also important because they activate killer T cells. So as you can see, helper T cells are really important in the actual fighting mechanism to destroy the antigens. Memory T cells are saved in the body to respond to the antigen just in case the body is infected by that very same antigen again. And suppressor T cells halt the immune response once the infection has been eradicated or if the immune response has become excessive. As you can see, T cells are rather critical in fighting infections, and if a person does not have functional T cells, then that person cannot effectively fight off infections. So I hope you can kind of see where I'm leading towards here when I talk about T cells and the importance of their functionality and how that might relate to HIV infection. But what I want you to keep in mind is that as we kind of backtrack a little and talk about HIV before we complete the loop and talk about how HIV affects the immune system is some of those basics about T cells and some of those basics about the immune response. Just to give you kind of another cool illustration that I found, this image is from AIDS.gov, a great website that has a lot of information about HIV and AIDS. You can see here that a healthy person has around 500 to 1,000 cells per millimeters cubed, and this is specifically referring to the number of T cells in the body, and this is what the normal level is. And 1,000 cells per millimeters cubed is a unit of measurement per certain area. And just to clarify, for our purposes, we're going to use T cells and CD4 cells interchangeably. If you really want to know what the small difference is, CD4 is actually the name of the receptor that is on the T cell surface that HIV uses to infect the cell. But we'll get into this a little later on when we talk about how HIV infection occurs. So now that we have a pretty good understanding of the immune system, let's go ahead and get into our primary focus. HIV stands for Human Immunodeficiency Virus, and as its name suggests, we can talk about 3 primary aspects about the virus. Firstly, you can only be the HIV virus can only be transmitted in humans. For now, we know that HIV cannot be transmitted in other animals. Secondly, it will weaken your immune system, and we'll get into this in a little bit, as well as about how exactly HIV affects the immune system. But the idea here is that it causes immunodeficiency. It will weaken your immune system. And thirdly, HIV is a virus. Viruses are non-living and infect a host cell in order to reproduce themselves. On the left side, I've provided a image of a microscope image of what HIV looks like under the microscope, and there's 4 HIV viruses that are in this image. But for a little bit more clarity, I've provided an illustration on the right side that shows all of the different key parts that are present both on the envelope of the virus, that outer coating, as well as on the inside. And we'll talk a little bit more in detail about this in just a little bit. But I wanted to point out just one key player, which is the RNA molecules, and that stands for ribonucleic acid. This is the primary genetic material that the virus contains, and this is what is injected into host cells in order to ensure infection will occur. But I'm going to get into all of this in just a little bit. I have a very good video that I'll show you all. So now, the next question is, how does somebody get infected with HIV? HIV is an infection, which means you actually have to get it from contact with somebody else. Body fluids are what transmit the infection. So I'm talking about blood, semen, pre-seminal fluids, rectal fluids, vaginal fluids, and breast milk. And so based on all of these body fluids, HIV can be transmitted through the means that are illustrated in this image, which I also found from AIDS.gov. Once again, fantastic resource. It really depends on the place that we're talking about in the world. In terms of the most prevalent cause of HIV transmission. But for the most part, the most common cause of HIV infection is unprotected sexual contact. However, there are a lot of other prominent causes of HIV infection: injection drug use, pregnancy, childbirth, and breastfeeding. Are also some of the causes. And when we talk about pregnancy, childbirth, and breastfeeding, that's where you might hear in the social media about mother-to-child transmission. So that's referring to this idea that an HIV-positive mother can transmit HIV to her unborn child. Another kind of less common causes are occupational exposure, typically by healthcare workers, and blood transfusions or organ transplantations. So once again, I want you to get a general picture of the causes of the transmission of HIV infection here. And we'll get a little bit more into detail about how HIV is transmitted in certain countries where we work in. So I found this really good video from the Howard Hughes Medical Institute that shows how HIV infection occurs, and I'm going to go ahead and show it to you guys. This is typically the HIV virus, and you can see inside that there are 2 RNA molecules that are found inside of the virus, inside of the envelope. There's also a molecule called reverse transcriptase, and I'll get into that in a little bit. But basically what happens is this virus travels and goes to the helper T cell, where it actually is able to make contact with the surface of the T cell through CD4 receptors. And you can see in this image, in this video, that these are the 2 red receptors that the virus is attaching to. And so that's why we can interchangeably call T cells and CD4 cells the same thing. So they attach to the CD4 receptors, and this causes a conformational change, not a transformational, a conformational change that ultimately leads to the fusion of the envelope, the outer coat of the virus, with the envelope of the T cell, and that causes the injection of those 2 RNA molecules, as you can see here. So you can see that there's the fusion occurring and then the injection of the RNA particles. And you can also see that the envelope proteins from the virus are now on the surface of the T cell. So then the virus protein coat breaks down, and you have those 2 RNA molecules with the blue reverse transcriptase on it. And so the reverse transcriptase is actually responsible for converting the RNA into DNA. Because if you'll remember from biology, the nucleus of a human cell actually contains DNA particles. And so we have to backtrack and convert that RNA into DNA. So that's what the reverse transcriptase does. And so it converts it into a single-stranded DNA molecule. And so you might actually remember from biology as well that the human body cell contains double-stranded DNA. So that single-stranded DNA that's been formed is going to go through the reverse transcriptase again, and it's going to become a double-stranded DNA molecule, which can now travel to the nucleus with the help of a molecule called integrase. And now it's traveling, going to the nucleus. Once in the nucleus, it locates a specific area of the host DNA, clips it, and actually attaches the viral DNA into the host DNA. You can see that this is a rather permanent form of infection. It's now incorporated into the DNA. And then you can have the transcription and the translation of that genome to produce the more RNA strands from that DNA double strand that's been incorporated into the human genome. And this transcription and translation process will create a variety of different molecules that can become proteins and basically go through the entire process of becoming entirely new viruses. So now once a new virus actually forms, it's able to be released from the T cell. And now you have hundreds, thousands, maybe even thousands, maybe even millions of different HIV molecules being released from this host cell to infect other T cells. And so basically, this infected T cell will eventually die out, and this causes a severe decrease in the T cell count. If you'll remember, the normal amount in human body is 500 to 1,000 cells per millimeters cubed, and that severely drops due to HIV infection. So I hope that that video gave you a pretty good understanding of the mechanism of action of HIV infection and how the genome of HIV can actually be incorporated into the genome of the host cell to cause a more widespread infection. Now, there are a number of different medications that scientists have developed, and they're continuing to develop, that are known as antiretroviral therapies. So the goal of antiretroviral therapies, which we also call ARTs for short, are to stop certain steps of that mechanism that we saw in the previous video so that HIV infection does not occur any further, so that more body cells are not infected. So we're kind of slowing the spread of HIV throughout the body. So if we stop one step of that mechanism, then we can essentially stop HIV infection from occurring in other cellular T cells. And so there are a number of different classes of ARTs that are available, and each class targets the specific set of the HIV infection mechanism. So I'm just going to go over 3 that I think are a little bit clear, a little bit easier to understand, and something that it's kind of good to carry with you. So one of them is a reverse transcriptase inhibitor. So you'll remember that reverse transcriptase is critical in transcribing the RNA strand that is present in the original HIV virus into a double-stranded DNA molecule that can be integrated into the host genome. But this medication prevents that reverse transcription from occurring, so it prevents that integration ultimately and the formation of that DNA molecule. So you can't integrate that DNA of the virus into the host DNA. Another is an entry inhibitor. There's a medication that actually prevents, really from the beginning, the attachment of HIV to CD4 cell receptors, and so that'll prevent HIV from entering the cell at all. Another one that I wanted to go over is an integrase inhibitor. And so remember, integrase is responsible for allowing the viral genome to be incorporated into the host genome. And so an integrase inhibitor is basically going to prevent that clipping of the host genome and that integration of the HIV genome. So you can see that from this image, once again from AIDS.gov, is there's a little bit of a summary of the benefits of antiretroviral therapies. They can help you live longer, they can lower your risk of developing non-HIV related illnesses, and they can actually reduce the chances that you're going to transmit HIV to others. And of course, there are some lifestyle choices that you can make to really reduce the effects of your HIV status. But really, what antiretroviral therapies do is they help to prevent AIDS. And so a lot of people don't really know that HIV and AIDS don't necessarily go hand in hand. Someone can be HIV positive without having AIDS, but somebody who does have AIDS was once infected by HIV. Basically, AIDS is acquired immunodeficiency syndrome. And so there's a couple of things that we should kind of discuss to give you an overview of what AIDS is. AIDS is the last step of HIV infection. If a person is not taking antiretroviral therapy, or if, for example, the virus has learned how to get around those antiretroviral therapies, viruses are very good about adapting to different circumstances, so they might become resistant to certain medications, but basically, if the HIV is allowed to continue to proliferate throughout the human body and cause destruction of T cells, remember those helper T cells, then the disease can progress into AIDS. People who have AIDS have less than 200 cells per millimeters cubed of the helper T cells. So if you remember from before, the normal is about 500 to 1,000 cells per millimeters cubed. And so you can see that there's a drastic reduction in the amount of helper T cells within the human body, and this in turn is going to decrease the body's ability to fight off infections. So AIDS is called a syndrome because it's not just one disease. It is a whole set of complications and issues that can develop as a result of prolonged HIV infection. And so now people who do have AIDS typically get diagnosed because they are HIV positive and they've had some sort of infection. There's a whole list of infections that doctors and hospitals use to diagnose individuals with AIDS. But you, of course, you have to be HIV positive and you have to be infected with at least one of a whole set list of infections that are possible. And so really the issue that happens when somebody has AIDS and basically develops their HIV positive status into AIDS is an increased risk of opportunistic infections. So when we say that somebody died because they had AIDS, we're kind of loosely using that understanding of the phrase. A person doesn't die from AIDS itself, but a person does die from the complications of AIDS, which is some sort of infection or some sort of disease that is caused due to the compromising of that individual's immune system. So there are a number of different actual causes of death for a person who does have AIDS, but we kind of often loosely say that a person dies of AIDS. And in the reality, like I said earlier, it's due to some sort of infection or condition related to their lack of immune system functionality. So I'm going to wrap up here by talking about some of the symptoms that HIV infection exhibits, as well as the prognosis for an individual who has been infected by HIV. We can actually break HIV infection into 3 stages. The first is an early infection stage. This is the stage during which a person has just been infected with HIV. They will exhibit flu-like symptoms like a fever, swollen glands, a sore throat, really typical flu symptoms, and really that's why it's a little bit difficult for people to get a diagnosis for HIV if they feel like maybe they just have a normal flu. So they don't need to go to a clinic to get tested for HIV. However, if a person believes that he or she participated in any activities that made him or her susceptible to HIV infection, then this is typically the time when that person gets tested for HIV. The next stage is actually a clinical latency stage. That really means a long-term asymptomatic period, which means that it's a period in which an individual does not experience any symptoms of HIV infection. Without antiretroviral therapy, this stage can last for about 10 years. However, with antiretroviral therapy, I actually did a little bit of research and tried to find out some information about this. It's been determined by the scientific community that with current antiretroviral therapies, if a person takes them on a regular basis and they don't experience a significant amount of complications in those therapies, then this clinical latency stage can last for several decades. So what I found is that if an individual is HIV positive and they take their medications on a regular basis, then they can actually live a lifespan that is equivalent to that of an individual who is not infected. So there's been some great advances in the scientific community recently in terms of getting those medications developed, administered to individuals who are HIV positive. But let's just say a person does not have access to those medications, as I talked about in the previous video, or if the antiretroviral therapies are just not working for that individual due to the virus adapting to the various classes of the drugs, then the final clinical stage is AIDS. And the initial onset of AIDS exhibits a weight loss, diarrhea, pneumonia, neurological disorders, and of course those opportunistic infections that I talked about earlier that can result in death. And so with that, I wanted to wrap up our webinar training video on the biology of HIV infection. I hope that wasn't too dense. I wanted to give you guys a good overview of the biology of HIV infection without nauseating you with a large amount of information. And so I hope that you've learned a lot about how HIV infection can occur, how it affects the immune system, as well as clinically understanding certain aspects of HIV infection and some treatment options. I wanted to thank AIDS.gov for much of the content of this video, as well as West One Services for that immune response image. So our email address is going to be in the comments below. Go ahead, if you can, subscribe to our video, and go ahead and watch some other of the videos that we have. If you're a volunteer, go ahead and move on to the next video. And you can also visit our website at www.pendulumglobal.org to learn more about our organization as a youth empowerment nonprofit and how you can get involved as a volunteer or make a donation. You can also go ahead to our Facebook page, like our page, www.facebook.com/pendulumusa, and you can also kind of see on a regular basis what we're up to. And so on behalf of Pendulum Global and on behalf of the entire team of our organization, I hope you enjoyed that video, and I'll see you in the next one.